What is the key difference between gram-positive and gram-negative bacteria? Imagine you are a detective trying to identify bacteria at a crime scene. One of your first clues is a simple stain test that reveals a lot about their structure. This test is called the gram stain, and it helps distinguish two main types of bacteria. But what exactly makes them different? That's what we're about to uncover. The main difference lies in their cell wall structure. Gram-positive bacteria have a very thick layer of peptidoglycan, which is a kind of mesh made of sugar and amino acids. This layer can be up to 20 times thicker than in gram-negative bacteria. They also lack an outer membrane, so their cell wall is exposed directly to the environment. On the other hand, gram-negative bacteria have a much thinner peptidoglycan layer, usually just a few layers thick. But they have an extra outer membrane that surrounds their cell wall. This outer membrane contains lipopolysaccharides, which are molecules made of fats and sugars. These lipopolysaccharides are important because they act as endotoxins and can trigger strong immune responses. When you perform a gram stain, the differences in cell wall structure determine how bacteria react. Gram-positive bacteria trap the crystal violet stain because of their thick peptidoglycan layer. They turn a deep purple or blue color under the microscope. Gram-negative bacteria, with their thinner peptidoglycan and outer membrane, do not hold onto the crystal violet after a washing step. Instead, they take up a counterstain called safranin, which makes them look pink or red. This difference is not just about color. It affects how bacteria respond to antibiotics. The outer membrane in gram-negative bacteria acts like a shield, making them more resistant to many drugs. Antibiotics like penicillin work better against gram-positive bacteria because they target the peptidoglycan layer directly. Another key point is how these bacteria produce toxins. Gram-negative bacteria releases endotoxins from their lipopolysaccharide outer membrane when they die or are damaged. These endotoxins can cause fever, inflammation, and even septic shock. Meanwhile, gram-positive bacteria produce exotoxins, which are secreted proteins that can damage tissues or interfere with nerve signals. Examples of gram-positive bacteria include Staphylococcus aureus, Streptococcus pyogenes, and Bacillus anthracis. For gram-negative bacteria, think of Escherichia coli, Salmonella species, and Pseudomonas aeruginosa. Knowing these differences helps doctors choose the right antibiotics and interpret lab results. It also guides infection control and treatment strategies. So next time you see a gram stain, remember that the key difference is in their cell wall structure. Thick peptidoglycan without an outer membrane versus a thin layer with an outer membrane containing lipopolysaccharides. That tiny difference makes a big impact in microbiology and medicine.